This is part 2 of a brief history of microbiology. So the final proof for the germ theory of disease came from a German scientist, Robert Koch, in 1876. So this is when they finally accepted that microorganisms are the cause of infectious diseases. So he used some steps that are called Koch's postulates to prove that the bacterium Bacillus anthracis is the cause of anthrax, which at the time was a big problem in livestock in Europe. So I do want you to know for the test, this would be great to know what Koch's postulates are used for. So make sure you know that they're used to prove that a particular microorganism causes a specific disease. So that's what they're being used for. So there are 4 Koch's postulates, and I want you to make sure you know what they are. I think it's just easier to take a look at a picture rather than just reading them off. So this is what Koch's postulates are. The first postulate is just saying that you should find the microorganism that causes the disease in the sick individuals, but you shouldn't find it in the healthy individuals. So that's the first postulate. The second postulate says that you should be able to isolate and grow the microorganism in a pure culture in the lab. So the reason you want to be able to do that is because if you have a bunch of different microorganisms, you want to be able to say that it's just this specific microorganism that's causing a disease than if you had a bunch of different ones. So you want to be able to isolate it and grow it in pure culture in the lab. So in the third step of Koch's postulates is you're going to take that microorganism that you've grown in pure culture and then introduce it into a healthy host, and they should get the same disease as the sick individual originally had. Okay, so you should be able to reproduce the disease. And then the fourth postulate just says that from the second sick host, you should be able to grow another pure culture of that same microorganism. So those are the 4 Koch's postulates. So make sure you know what those are. And we actually will be coming back to these again later in this semester. So just make sure you learn them now because they'll come back. Okay, and then some other developments in microbiology have to do with vaccination. Just to introduce some terms, immunity just means that you're protected from disease, and obviously a vaccine will help do that for you. Edward Jenner was an English physician who in 1796 became famous because he came up with the very first vaccine. And the vaccine that he developed was against this disease, which is pictured on the right, which I don't know if any of you can guess what that disease might be, but it's for smallpox. So he came up with the very first vaccine, and it was to protect people against smallpox. What he used was cowpox, and cowpox would just give people a very mild pustules on their skin. It wouldn't make them very sick at all, but then it would protect them from smallpox because it looked very similar to smallpox to the immune system. And then people would be protected for the rest of their life. So he came up with the very first vaccine. Some other developments with vaccines. Louis Pasteur is back. He was a very busy guy. He discovered something with vaccines called attenuation. The word attenuate just means to weaken. And he discovered attenuation quite by accident. So he was working on developing some vaccines, and one that he was working on was for chicken cholera, which is caused by a particular type of bacteria. He was growing the chicken cholera bacteria in his lab, And it was summer and time for summer vacations, so he went on vacation, came back a couple weeks later, and the cultures that he had growing of the bacteria were pretty old, and they've been running out of food and they weren't in the best living conditions. But he's like, well, I'll just give those to the chickens and see how it works. So he gave the old bacteria to the chickens, expected them to develop chicken cholera and get sick, but they didn't. So that surprised him. So then he went and got fresh bacteria, gave that to the chickens, and they didn't get sick. And he's like, oh, I'm onto something. And so he discovered that by growing these microorganisms in bad living conditions, or not the ideal living conditions, they would weaken or attenuate. And then that would make it so the individual's immune system could then fight them off much easier, and they'd develop immunity and wouldn't get sick. And so he developed vaccines that way for chicken cholera, anthrax, and also for rabies by using this method. We also had the development of chemotherapy. And when we talk about chemotherapy in microbiology, we're not talking about treating cancer. We're talking about treating infectious diseases by using antimicrobial drugs. We do differentiate between antibiotics and synthetic drugs, although I'm not going to ask you the difference between them. But there is a little bit of a difference. Antibiotics are actually naturally produced by bacteria and fungi like mold to actually kill off their neighbors, because they're competing for living space and food. And if they can kill off their neighbors, then they can survive better. And so that's where antibiotics actually come from. And so then we just gather those and then give those to the patients. And then we also have synthetic antibiotics as well, which are man-made. The synthetic antibiotics would be, for example, the sulfa antibiotics. So the brand names with those you might be familiar with would be like Bactrim and Septra. Okay, the very first antimicrobial drug that was ever used was developed by a German scientist named Paul Ehrlich in 1910, and he discovered a drug called salvarsan. And it was made from an arsenic derivative, and arsenic is very toxic, it's a poison, and he used it to cure syphilis. Syphilis is caused by a bacteria called Treponema pallidum, which is pictured over here on the bottom right. And prior to 1910, if you were infected with syphilis, we didn't have anything to treat you with. But they would try to treat you with things like mercury, which didn't cure you, but would probably make you pretty sick because mercury was pretty toxic. But this was the first effective treatment against a microbial infection. So this is what they used until penicillin came out in the 1940s. So that was our very first antimicrobial drug. The first antibiotic that was ever discovered is penicillin. And it was actually first discovered in the late 1800s by a French scientist, but then nobody really paid attention to it. And then Alexander Fleming, a Scottish scientist, accidentally rediscovered it in 1928. He was working with the bacteria Staph aureus, and fortunately, in his lab, he was not very clean. He tended to let his petri dishes stack up by the sink, and he didn't do his dishes very often. And it was summer and it was time to go on vacation again. So it's kind of a common theme here. So he was gone for a couple weeks and came back and went to do his dishes. And he noticed on one of the petri dishes that had been sitting out without the lid on it that some mold had landed on the petri dish. And around the mold, the Staph aureus bacteria had been killed off. So the mold is this white circular spot, and you can see that it's clear around it. So the mold had secreted something that had killed off the Staph aureus bacteria around it. And so Alexander Fleming was really excited about that because he was like, that is fantastic. He's like, anything that can kill Staph aureus is just amazing. And so he was able to isolate penicillin from this mold called Penicillium chrysogenum, but he wasn't a chemist, and so he wasn't able to isolate enough of the penicillin to actually test it on mice or anything to see if it actually would cure a bacterial infection. So to fill in our blank here, we'll put in that the penicillin killed Staph aureus. And so we actually had to wait until the early 1940s to some other scientists who discovered Alexander Fleming's paper about penicillin, and they were chemists and were able to isolate enough penicillin to actually test it and see that it actually would cure bacterial infections. There's lots of different branches of microbiology, so lots of different areas that people can go into. We have bacteriology, which is the study of bacteria. We have mycology, which anytime you see that prefix with MYC, that just means fungi. So MYC refers to fungi. So mycology would be the study of fungi. So you would be studying the yeast and mold there. And then virology would be the study of viruses. Immunology would be the study of the immune system. Medical microbiology, you would be studying microorganisms that cause infectious diseases. Industrial microbiology, you would be studying microorganisms and how you could use them to produce things. So maybe you would be looking at how you could use them to produce fuels, or maybe you looking at enzymes that they make to maybe use to make different dyes, or maybe clean up, toxic waste. So there's lots of different uses for microorganisms in industry. Maybe you're wanting to use them to make different drugs. So there's lots of industrial uses. And then the last one, microbial ecology. So some of you may have taken an ecology class, and this is just ecology on a smaller scale. So this one is just the study of how microorganisms interact with each other and their environment. Okay, so that's microbial ecology. So you're just looking at how the microorganisms interact with each other. Maybe one microorganism is producing a product that another one needs, or maybe one is producing something that kills off another species. and then how they're interacting with their environment, because they can change their environment. Maybe they're making it more acidic, so they can change their environment as well. And the last development to talk about is recombinant DNA, which has come about because of genetic engineering. So what recombinant DNA is if you just look at the word recombine, you're just going to take two different pieces of DNA coming from two different organisms and join them together. So a good example of this is how we make human insulin. So what we do is we take out the insulin gene from a human cell, and then we're going to take that human insulin gene and stick it into the bacterial chromosome. And now we've got recombinant DNA. So we've recombined the human insulin gene into the bacterial chromosome. And now we can grow that E. coli bacteria in the millions in the lab, and the bacteria will read the human insulin gene and make human insulin for us. And then we can collect that and package it up and then give it to our diabetics. The other microorganism that's also used in this way to make human insulin is yeast. And we just do the same thing with it. So either yeast or E. coli can be used to make human insulin. And there's a lot of other human drugs that are made using this process. And so we'll be talking about some other uses of recombinant DNA a little bit later in the semester too.